Welcome back everyone. So in this video, I will be introducing you to the different types of evidence for evolution. Now, essentially, evidence of evolution is pretty much anything that can demonstrate a change in characteristics of an organism, a species from the ancestors. So anything that demonstrate a change over time can be considered evidence for evolution or of evolution. The first type of evidence is fossil record. Now, a fossil, I'm sure you have heard of the word fossil before, but a fossil is the preserved remains or trace of any organism from the remote past. So there are two types of fossil. You can have the preserved remains or you can have trace fossil. Now, preserved remains would be something like this. So when you have most of the time hard parts of the body, such as teeth and bone and shells, and they were reserved and because they were buried underground under high pressure, and then you would have something like that. So most of the time you will see preserved remains as a skeleton, for example, of the organism in this case. So this is preserved fossil. There is actually another type of fossil, which is this one, which is trace fossil. So this is not an actual part of the organism, but it is, in this case, an impression on an imprint, and it could provide evidence of, say, the pores or certain parts of the organism. You could also have trace of the entire organism if you have a really small organism. So you have preserved fossil or trace fossil, two types. Okay, now fossil record is essentially the total collection of fossils discovered and undiscovered, and the record of fossil can show that over time changes have occurred in the features of living organisms. So that's why fossil record is probably the most common type of evidence of evolution that people think of when people think of evolution, because it could tell you that in the past organisms used to look like this and now it looks something else, and that was because of a change in selection pressure. In order to successfully use fossil to provide evidence for evolution, what we normally have to do is to date the fossil, which just means that you try to determine the age of the fossils. So the process of fossil dating is essentially determining the age of a fossil. There are two methods for fossil dating, relative dating or absolute dating. So relative dating is when you compare the placement of fossils in layers of rocks to determine which fossils are older. And the general rule of thumb is anything that is in the lower layer is going to be older. So I'm giving you an example here. So let's say, for example, I dig up a rocks and there are multiple layers. The one that is at the bottom, this one, it's going to be the oldest. So I think it makes sense. The oldest has to be the first one that laid down, and then so on and so forth. So the oldest one is at the bottom, and which means that all of these fossils at the bottom or in the lowest layer of the rocks would be older than the fossils in the upper layer. So as you go up, the age of the fossil is going to be smaller and smaller, if you will, and then you have that something which would look like a human skull, and that would be a relatively new fossil compared to those in the bottom layer. So this is newest. The second type of fossil dating is called absolute dating. And the difference between relative dating and absolute dating is you go back to relative dating, it allows you to compare. So if I have two fossils in the same rock, I can tell which one is older, but I cannot find the actual age of the fossil using relative dating. If you want to know exactly how old the fossil is, like is it 50,000 years ago or 500 years ago or what, you need to use absolute dating. So it will determine the exact age of a fossil by detecting the decrease in the amount of carbon-14 atoms in the fossil over time. Now, just a quick recap, this is basically the mass number. So carbon-14 is a type of carbon atom that contains 8 neutrons and 6 protons for a mass number of 14. And it is pretty unstable. And we don't have to go into too much of the chemistry behind this, but basically carbon-14 would decay into carbon-12, which is the carbon that we commonly see over time. And what we're going to use is the half-life of carbon-14, and the half-life of it is 5,700 years. So for every 5,700 years, 50% of carbon-14, which is half-life, is going to become carbon-12. And you can use this information to date the fossil, because you can compare the percentage of carbon-14 that remains in the fossil. So for example, if I find that, so this is just the half-life, you don't have to worry too much about this, and they actually do compare it to nitrogen-14 just to work out what the half-life is. So if I have, say, 25% of carbon-14 left, this means that I have gone through 2 half-lives, because so in the first one, the first half-life, is when you go from 100% to 50%, and then the second half-life is when you go from 50% to 25%. So if I analyze the sample and found that there is only 25% of carbon-14 left, and you normally compare it to the nitrogen-14 in the sample, which is why the graph is here. So if I only have 25% of carbon-14 left, it means that 10,000 and 10,000 and okay, I can't do maths. Anyway, the first one's 5,700 years, and the second one's 5,700 years as well. So this is good. 11,400 years have passed, and that would be the date of the fossil. So that's absolute dating. And the interesting thing about fossil record is it allows you to really track the change of organisms over time. So, for example, I have the sequence of the animal kingdom. So we know that we started with shelled invertebrates, and then they had a vertebrate. That you get fish, we had your spy, and then we evolved into amphibians, and then reptile, and then mammals and birds and men. And then you can see a lot of fossils that supported this theories. For example, this is a fossil of an organism called Tiktaalik, and you could see that it looked like a fish, but it had legs. So this is evidence for the fact that we started under the sea. So we started as aquatic animal, and then at one point, one of these organisms made it to land. And then you have another one, such as this one. I'm not even going to attempt to say this name, but it's like a half bird, half dinosaur kind of organism, and it show the evolution from reptile to bird. Or you can have this one, Ida, which show the evolution from mammals to primates, if you will. So you can see some really interesting in these are called transitional fossils, when they had features of both the ancestor and then the newly evolved species. The next type of evidence for evolution is what is called comparative embryology. And what it is the study of the growing embryo in plants and animals. So what you do is you look at the embryos of different organisms at different stages, and you compare and identify similarities and differences between them. And it tends to show that closely related organisms would go through similar stages of development. So I'm just going to show you a photo of the embryos, I think the first few days of development of different organisms. So we'll go from, say, fish, say chicken, to rabbit, to human, and you can see in the first few days the embryo of vastly different organisms are actually very, very similar. Generally, you can also see that there are two features. So the first one is here. These folds are called gill slits, and they are non-functional. So in a fish, these gills would eventually develop into actual gills. And in most of these organisms, except for the first two, which are actual fish, they will eventually be removed or develop into something else. But in the first few days of the embryo, you still see them, even though they no longer functional in terrestrial organisms. And you can also see that many vertebrates demonstrate a primitive tail at certain stage. So human also has this part, which is like the tail, even though we don't have a tail, last time I checked. So it is amazing that vastly different organisms can have very, very similar embryo at the start and then eventually develop into different organisms. The other type of evidence for evolution is comparative anatomy. Now, comparative anatomy is the comparison of the anatomy of different species. So you don't look specifically at the embryo, you look at the species once it is fully developed. And generally, when you look at the anatomy of different species and compare them, there are two types of structures that you can identify. The first one is what is called homologous structures, and the second one is analogous structures. So let's just look at the differences between these two. So homologous structures are structures which have similar anatomy but have evolved into different functions. So they evolve into a variety of distinct forms with different functions, but the underlying anatomy is similar. So similar anatomy, different functions. And this indicates that these organisms once upon a time have shared a common ancestor, but then eventually, because they were subjected to different selection pressures, they have diverged into different organisms. So an example of that is if you look at the limbs of, again, a range of organisms from human to dog to horse to bird to seal, you can see some very similar underlying anatomies, but obviously the actual function has changed. So this is an arm of a human, and it has a very different function from the legs of horses, and very different from wings of birds or the seal. So very, very different functions, but the underlying anatomy is the same. So these are homologous structures. Um, Another example of homologous structure is what is called vestigial organs, and basically they are remnants of organs that used to be functional in the past in our ancestors, but we no longer use them, but we still have those structures. So anything you can think of that are there but doesn't have a function, that's a vestigial organ. And the classic example is in a whale, which has this, which is this is the pelvis bone, because whale used to be a terrestrial animal, which means that it used to be on land, and then it decided to go back to the sea, but it still has the hip bone, if you will, even though obviously it doesn't need it anymore. The other types of structures that you can identify by comparing the anatomy of different organisms is what is called analogous structures. This is when they have similar functions, but with different underlying anatomies. So it indicates that these organisms were subjected to similar selection pressure, even though they didn't share common ancestors. So they have similar, see the difference between homologous and analogous. Homologous is when they have similar underlying anatomy but different functions. So they start off with a common ancestor and then they diverges into different organisms. Analogous structure is when you have different organisms, so they start with different um, ancestors, but they have similar selection pressure. So that you probably find these organisms in similar location or locations with similar conditions. So an example of analogous structures would be, let's say you look at with fin and wing and flippers, which have very similar functions, but the underlying anatomies are very different. Or you have the wing of a fly versus the wing of a bat. They both to fly, but very, very different structures. The next type of evidence that we can consider is what is called biogeography or geography sometimes. And basically, we're focusing on the distribution of life forms or organisms over geographical areas, both in the past and the present. And normally, what you will see when you look at the locations of the organisms in the past and now is that closely related species tend to be close in physical proximity to one another, which means that they were subjected to similar selection pressures. So, for example, most modern marsupials, so they're very, very similar to each other. If they're all marsupials, most of them are found in Australia. That is an indication that, especially because Australia is an island as well, that they have very unique selection pressures, and that causes or has caused multiple species to evolve in the same way. Some of them are actually in another part of the world, but the majority is in Australia.
Some exceptions can be explained due to continental drift, and this is actually quite interesting. If you look at this native Australian plant, the Waratahs, you can find it in Australia, but you can also find it in South America or Indochina or parts of Africa. And the reason for that is because of continental drift. So these continents used to be very close to each other, and that's how the plant could spread. And then once the continents broke apart, you could find the plants in multiple parts of the world. The very last types of evidence is molecular and genetic evidence, and this is relatively new compared to the other types of evidence, and we are gaining more information from this because of the advance of technologies in the past few years. So essentially what it is you want to identify conservation of DNA and protein sequences as a basis for determining evolutionary relationships. In general, if two species are very closely related, their DNA and protein sequences will be very similar to each other. So this is an example. Chimpanzee is 99.5% similar to the DNA of human, which I think it's higher than what most people expect. But even if you think of the fruit fly, 60% similar DNA to a human. So I think molecular evidence is probably the most accurate in allowing you to know which two organisms are more closely related to each other.